Imagine you're a medical student cramming for the USMLE Step 1 and you realize DNA is packed so tightly in the nucleus that it's like fitting 2 miles of thread into a tennis ball. Mind-blowing, right? Hi, today we are diving into two high-yield USMLE Step 1 concepts about chromatin structure that you need to know for test day. Let's break it down in the upcoming slides. First up, let's talk about how DNA fits into the nucleus, because that's a Step 1 favorite. Check out. It's all about the nucleosome. Picture this as beads on a string. DNA loops twice around a histone octamer made of two copies each of histones H2A, H2B, H3, and H4. That's the bead. Then histone H1 comes in like a stabilizer, binding to the nucleosome and the linker DNA to form a tight chromatin fiber. This packaging is crucial for fitting DNA into the nucleus and regulating gene access. Now let's zoom into the chemistry. A classic Step 1 test point. DNA has a negative charge because of its phosphate groups, while histones are positively charged thanks to amino acids like lysine and arginine. Opposite attracts, right? That's why DNA and histones sticks together so well. For the USMLE, remember, nucleosomes are the basic unit of DNA packaging, and they are critical for chromatin stability and gene regulation. If you see a question about histone modification or chromatin structure, this is your foundation. Moving on to the next slide. During mitosis, chromatin condenses even further into the chromosome to prepare for cell division. But when does DNA and histone synthesis happen? That's right, during the S phase of the cell cycle. Step 1 loves to test this, so lock it in. S phase is for synthesis, M phase is for mitosis. Now here's the curveball. Mitochondrial DNA. Unlike nuclear DNA, mitochondrial DNA is circular and doesn't use histones. It's independent, like a little genetic island in the cell. This distinction is huge for Step 1. Nuclear DNA is linear, histone bound, and condenses in mitosis, while mitochondrial DNA is circular and histone free. The USMLE key point here. Know the difference. They might ask you to compare nuclear and mitochondrial DNA or tie this to a clinical scenario like mitochondrial disease. Either way, you're ready. And there you have it. Two high-yield USMLE Step 1 concepts on chromatin structure in just under 3 minutes. From nucleosome to mitochondrial DNA, you are now armed with the basic to tackle those tricky questions. If you found this helpful, give this video a like, subscribe for more USMLE prep, and let me know in the comments what topic you want next. Let's ace Step 1 together. See you in the next video.